Okay, so in terms of the peptidoglycan layer, so comparing here the gram-positive and the gram-negative cell wall, so peptidoglycan layer of the gram-positive bacteria would have a very thick na peptidoglycan layer. It's made up only of one layer. For the gram-negative, on the other hand, so its cell wall made up of two membranes. One is the inner membrane, it's primarily made up of a thin layer of the peptidoglycan. Another one in the case of your gram-positive cell wall, the cell wall here may naka-attach. So basically, it's made up of the teichoic acid attached to the cell wall of the gram-positive cell wall. And then we have also here the lipoteichoic acid, which is also attached here to the plasma membrane of your gram-positive. Teichoic acid and the lipoteichoic acid are all absent in your gram-negative bacteria. Okay, now we go to your gram-negative bacteria or gram-negative cell wall. So again, the gram-negative cell wall is made up of two layers. So we have here, this is your cell membrane, may nakakoat dito. This is your inner membrane, which is made up of a thin layer of the peptidoglycan. Then meron dyan sa gitna space. And we have here your outer membrane. So in between your peptidoglycan, the inner and the outer membrane in your gram-negative cell wall, may periplasmic space. You call it once your periplasmic space. Okay, so periplasmic space is a gel-like matrix primarily made up of the nutrient-binding protein plus enzymes involved in the degradation and detoxification. Periplasmic space could not be found among your gram-positive cell wall kasi isa lang naman ang membrane ng ating gram-positive cell wall. Unlike in your gram-negative, may dalawa siyang membrane. And each of these two membranes here are separated from one another by the periplasmic space. Okay, now we go to the outer membrane of your gram-negative cell wall. The outer membrane of your gram-negative cell is made up of the phospholipids, protein, and another substance, LPS, lipopolysaccharides. The lipopolysaccharides here are divided into three layers. Number one, we have here the somatic or the O polysaccharide. The inner one is your core polysaccharide. And then we have here, or the middle one is your core polysaccharide. And another layer is made up of the lipid A, or you call it once as the endotoxin. Again, all this one are found on the outer membrane, specifically on the polysaccharide layer of the outer membrane of your gram-negative cell wall. Okay, now we go to the lipopolysaccharide layer of the outer membrane of your gram-negative bacteria or gram-negative bacteria. So again, it's made up of the layer somatic O polysaccharides. So the somatic O polysaccharides forms part here as the O antigen or somatic antigen of your bacteria. Okay, so the somatic antigen could be identified here by serological typing by the use of your anti-sera, like your anti-O, which is your antibody. So, parang blood typing lang siya. Gagamit ka nitong reagent na to, then you have your bacterial suspension, tapos i-mix mo lang siya. Pag nagkaroon ng turbidity or agglutination, so meaning to say your bacteria contains the somatic or the O antigen. And that's pertaining here to the somatic polysaccharide layer of its lipopolysaccharide. Example for that na may somatic antigen, we have here your Escherichia coli, O15787. This is a strain of your Escherichia coli having that somatic antigen. Then we have here another layer, very important layer. In the LPS, we have here your endotoxin, or you call that one as your lipid A. So the endotoxin or the lipid A moiety here is actually responsible here for the manifestation of the fever and the shock here if the patient has been infected by a gram-negative bacteria. Okay, then we have the function of your outer membrane of your gram-negative cell wall. The first one, it serves here as being a membrane to try to regulate the entry of the hydrophobic and the harmful substances in and out of the cell. Another one, it would also allow here the entry or the passage of the water-soluble molecules here by passing through pore-like structure or canal-like structure. You call this one as porins. Another one, ang membrane din na ito, the outer membrane, is also used by the bacteria in order for that to attach to the tissue of the host here in order for them to invade and to infect the host. Okay, then we have here bacteria, which is would have here, could not really classify as a gram-positive or gram-negative. Okay, you call that one, sir, acid-fast na bacteria. Okay, the acid-fast bacteria, that includes your mycobacteria, mycobacterium, and we have also your nocardia. So they are acid-fast basically because their cell wall is made up of very thick na lipid layer or waxy layer. You call that one as your mycolic acid. When we are doing the gram stain, ang ginagamit na decolorizer is the acetone. So kaya pag gumamit ka ng acetone, pag gram negative ito, pwede niyang butasin ang cell wall ni gram negative but not your gram positive. The same through with your acid fast. Hindi niya kaya. So instead of using here the acetone as decolorizer, we are using here the acid alcohol. So for you then to identify your acid fast bacteria. Pag gram stain ang gagamitin mo na method here, ang bacteria mo ay like if you have the Mycobacterium tuberculosis na bacteria, magiging gram positive ito. Okay, so eventually, but of course, the appropriate na staining technique will be the acid-fast staining dapat. Then we have here the bacteria Mycoplasma and Ureaplasma. So this is a special type of bacteria wherein they don't have the cell wall. So at birth, okay, wala talaga silang cell wall. We need to differentiate your Mycoplasma and Ureaplasma with your L forms. L forms are bacteria wherein they lost their cell wall because of exposure to environmental factors. Pero beforehand, meron pa silang cell wall na sira lang. Magkaiba sila. Mycobacteria, wala talaga, Mycoplasma, Ureaplasma, wala talaga silang cell wall. Ang mga L forms, dati may cell wall, nasira nga lang because of exposure to some factors, environmental factors. So why you need to differentiate one another because both of them wala silang cell wall. Okay, so we have here the differentiation between your gram-positive and gram-negative envelope here. So we have the peptidoglycan. So again, the peptidoglycan and the gram-positive here would be a very thick na layer compared with the thin inner membrane peptidoglycan layer for the gram-negative cell wall. And then another one, the gram-positive would have here the presence of your teichoic and your lipoteichoic acids which are absent among your gram-negative. Periplasmic space, since dalawang layer ang ating gram-negative, meron siyang outer membrane plus meron siyang periplasmic space which are all absent among your gram-positive cell wall. Then we have here the lipid content, so madami. Mas thicker na lipid layer makikita sa gram-negative compared to your gram-positive. So protein, much of the protein polysaccharide layer ang ating gram-positive compared with the high lipid na layer found in your gram-negative. Okay, now we go to the surface polymers, or you call that one, sir, appendages. So there are some bacteria wherein they have surface appendages that might eventually found here on the outer membrane attached to that one. The first one, we have here your glycocalyx. So the glycocalyx here try to surround the cell, and at the same time, this one is made up of polysaccharide, polypeptide, or even either of that one. Your glycocalyx here may be in the form of your capsule, which is considered to be a highly organized layer, plus that one is firmly attached to the cell membrane. Whereas your slime layer, on the other hand, that one is unorganized layer, and that one is not firmly or loose. This one is just loosely attached to the cell membrane. So example bacteria which has this capsule, we have here Bacillus anthracis, Klebsiella pneumoniae, and we have also here your Streptococcus pneumoniae. Okay, the main function of the capsule here, so since may extra layer pa siya after the cell wall, ang nangyari ay this is one of the virulence factor of your bacteria. So that would allow them to escape for the recognition of your phagocytes. Since pa may nakakoat sa kanila, nakatago sila sa kanilang capsule, hindi sila makilala ni phagocyte. Remember that your phagocyte are those that try to kill the pathogen inside your body. Pag hindi ito makilala ni phagocytes, so mangyayari, hindi sila mapapatay. And therefore, hindi ka gagaling sa sakit if you have been infected by this one. So, making this one as their virulence factor. Okay, the capsule here may be in the form of your K antigen or the VI antigen. So, capsule pa din ito. The VI antigen, most likely kasi capsule here is the capsular antigen is the K antigen. The VI antigen is very specific siya na capsular antigen found in your Salmonella typhosa. So, like for example, if your bacteria, meron siyang somatic or the O antigen, ang somatic or the O antigen, nasa pinakalabas talaga siya. Then, we have your capsular here. Kung ang bacteria mo, like for example, may somatic antigen sa capsular antigen, when you wanted to identify that one by your serological testing, again, pag serological testing, you are using here anti-sera. So, kung somatic ang minimeasure, dinedetermine natin, you are using anti-O, anti-sera. Pag capsular is anti-K na anti-sera. So, again, if your bacteria would have the somatic, meron pa siyang capsule. But if you wanted to know here, wanted to determine the presence of the capsule, dapat muna sisirain mo si somatic antigen para lumabas si capsule. It's because here, pag hindi mo siya sinira si somatic, it try to mask the presence of your capsule here, so it will give a negative result when you're having the serological testing. So, when you're doing the serological testing, ang procedure natin here is to heat first your bacterial suspension as your sample in order for you to destroy the somatic antigen para ma-expose the capsular antigen. So, if your bacteria, for example, will have the capsular antigen, it will really expose and it will give a positive reaction with your serological testing. Okay, the next appendages here, we have here your flagella. So, flagella here is a protein filaments which try to be extending here from pole here, one pole or one part of the body of the, or it depends on several parts of the body of the bacteria. Okay, so the flagella is actually responsible here for the motility of your bacteria. So we are expecting here that all your bacteria which are motile, they have the flagella. Okay, so again, if we try to discuss each of the bacteria naman, we will identify they have a very characteristic na mga motility. So again, if they are motile, assume that they have the flagella. Classification of the flagella here according to the location or the arrangement of the flagella. So first one, monotrichous. Monotrichous here characterized by only one flagella at one end. Lophotrichous on the other hand, so characterized here by two or more flagella found only in one pole end. Sa isang side lang. Pag amphitrichous here, so there are two, one flagella on one end, another flagella here on another end. On the opposite side, they become here the amphitrichous. Pag peritrichous, so the type of the flagellar arrangement here wherein the entire body of the cell here contains, or they will have here projecting na flagella, the entire body of that. That'll become here the peritrichous. So example of the bacteria which has the flagella, we have here the Proteus. The Proteus here would have a very characteristic na swarming motility. So yan ang tawag sa swarming motility because of its peritrichous na flagella. Okay, for the antigen, we consider here the flagella as the H antigen or the flagellar antigen. Again, O for the somatic, K for the capsular, and H for the flagellar antigen. And this is also identified here by serological typing, serological testing with the use of your anti-H, anti-sera, just like your anti-O and anti-K. So again, pag serological testing, you could identify, differentiate, classify them that become a serovariety. Diba? Serovar is based on serological differences, serological testing result differences. Another appendages, we have here the Fimbriae and the Pili. The Pili and the Fimbriae are actually extension din siya, but they are not the same as the Flagella. This one would be shorter, and this one would have here much thinner compared to your Flagella. The Fimbriae, this is other name for the common Pili. So basically, that's one you could find here approximately few to hundred Fimbriae to each or projection for each of the cell here. So the main function of the Fimbriae is to use this one by the bacteria to attach to one another or even allowing them to attach to the surface or the tissues here of the host in order for them to invade and to cause infection. And as we have here the pili, this pertains to your conjugation pili or your sex pilus. So from the word itself, this is a holotrope here that would enable here two bacteria to exchange. Ginagamit ito for their conjugation. It's a process of their genetic exchange. So kung ito ang bacteria mo, itong bacteria mo, para may tube dito. Okay, so dito dumadaan, that would, para siyang conjugation, or mating. So that would allow them to exchange their genetic materials. Okay, and lastly, we have here the another appendages. We have here your axial filaments, also called the endoflagella or the periplasmic flagella. So this is a type of flagella here. So it's a bundle of the fibrils usually attached in one pole end of your spirochetes na mga bacteria. Spirochetes are spiral na bacteria, and there are three genus only of the bacteria belonging to the spiral or spirochetes. So includes here the genus Treponema, the genus Borrelia, and we have your genus Leptospira. And this is responsible here for the spiral motility of this genus of the bacteria. Okay, so thank you.